Welcome to the Infinite and Beyond, the MIT Alumni Show. I'm your trusted host, Rob Kalwarowski, MIT Class of 2010, world-renowned executive coach, author, and speaker. Each week, we are sharing the stories with our friends and classmates, fellow MIT alumni who've taken action to shape the world. We'll share what we've learned, top strategies, and bring our community together to help everyone achieve our goals. No, this show is not officially affiliated with MIT, so any views expressed on this show are of the host and guest only. This show is designed to build a community that trusts, connects, and shares with each other so we can all take action towards a better world. Let's get rolling. Welcome to the Infinite and Beyond, the MIT Alumni Show. Of course, I'm Rob Kalwarowski, and we have a special guest with us. Obviously, another MIT 2010. Ryan Flynn is with us. Ryan, how are you?
Good. Nice to talk to you, Rob. Doing all right on this Friday.
Amazing. Yeah, Ryan, thanks for joining us today. Of course, for folks out there, Ryan, he's an MD, he's a PhD. He's the assistant professor of stem cell and regenerative biology at Harvard. At Harvard. But he's also the principal investigator of the stem cell program at Boston Children's Hospital. So we have another amazing healthcare guest. Now, Ryan, I'd love to ask you, can you tell us a little bit about how you got from graduation in 2010 all the way to where you are today?
Yeah, of course. And I have an affiliation at the Broad, so yeah, that's a Harvard MIT, affiliation. And actually, fast forwarding, my wife is an assistant professor at MIT, so I've made sure to keep my ties there.
Thank goodness.
But yeah, no, I was course 7, graduating, and basically went straight away into grad school. So I ended up, I actually had a vision that I would be a surgeon, and thought I needed to go to medical school. Really liked doing research, though. I worked in Phil Sharp's lab while I was at MIT. So thought, all right, I got to do MD work, and I'd like to do research. I was like, MD PhD sounds like a good plan, and ended up going to Stanford as an MD only, actually, to start. Immediately knew I needed to do a PhD, so started rotating in this sort of thing. But basically realized that probably clinical medicine wasn't for me. And really liked the research side of things quite early on, and perhaps even in the number of like single-digit weeks of arriving at Stanford. I knew that, but basically spent a lot of time at Stanford. So I ended up doing my MD and PhD and postdoc at Stanford. So didn't really go anywhere for basically a decade, 2010 to 2020. But yeah, I did my PhD there, did a lot of molecular biology and RNA biology. After I graduated through the MD, went to do my postdoc. I basically moved a couple buildings over to work in a chemical biology lab. My mentor there was Carolyn Bertozzi. And then as I was, wanting to run my own lab, after that, did a search and, didn't realize that I'd be applying during a pandemic, but I went up, I put my application in the fall of 2019, thinking nothing of that, future. And so ended up with a very narrow set of options, but ended up having a great opportunity to come back to Boston and start my lab here at Children's Hospital. So my lab is physically at the Children's Hospital, and I do teaching, and my faculty appointment is also through Harvard, again, University. And moved here about 4 years ago, moved back here 4 years ago. And still continuing along the research trajectory, a lot of RNA biology, but, we can talk about that later. But that's sort of the trajectory, I would say. Turns out to be fairly simple in terms of locations, but took a long time.
Yeah, I mean, of course, the higher education's a long ride. Now, Ryan, can you tell us a little bit about your research? Like, obviously, it's in stem cells, but what are they for, and what do they do, and what are you trying to learn?
Yeah, so my sort of training, I would say, took place in a couple of different domains. So I was trained a lot in basic biology, basic biological research. So very mechanistic, sort of always just asking the sort of random questions of why, not even how, but like really just why is this happening? What is going on? The domains that I've studied are broadly in RNA biology and glycobiology. RNA is the messenger. It's what's communicating all that information. Glycans are this actually really cool other polymer that I didn't really know about until I got to my postdoc. And they're what decorate the outside of cells. It's actually how cells talk to each other. It's how cells differentiate between hosts and pathogens. The way we recognize viruses and bacteria is through these glycan polymers, these sugar polymers. And when I was a postdoc, I sort of found a way to bring those two fields together. And I'm sort of now studying the interface of RNA biology and glycobiology. And so the lab now is really thinking about the activity of RNA outside of cells. So we've been looking at mostly mammalian systems. I did do that MD, so I do care about human biology and human disease. And so we think a lot about human systems, cancer, autoimmunity, these kinds of things. And so we study the molecular events of human cells and the outside layer of how RNA can contribute to cells talking to one another, and how pathogens interface with us. And especially thinking a lot about how the immune system can recognize new molecules on the cell surface, and that those molecules are what we're studying.
And so I haven't done biology since 7013.
Yeah.
So the molecules on the outside, those are not RNA, right? Like
Classically,
inside it, right?
Exactly. Yeah, that's exactly right. So this is the excitement that we have, right? So the traditional models of biology would tell you that proteins are outside of the cell. You have a lipid bilayer that holds everything in, that there are these carbohydrates, these glycans that stick out like little antennae that are signals, but that inside the cell, nucleic acids are really popular, RNA, DNA, and this is where you have all of your transcription and translation and replication and all this stuff. And so then the sort of exciting bit for us is that we actually found, if you look maybe up here, that there's this little RNA thing that's sticking out actually on the surface of the cell.
Wow.
And so we've been trying to understand, why do cells put specific RNAs outside? What are they doing? How do they get there? Are they different in disease? But it's a very different model of how a cell is organized, how the body is set up, right? And so, there's a lot of mechanistic questions that's what I like thinking about. But the kind of really cool thing, I think, is that because we are on the outside of the cell, this is all where the drugs work. This is all where the diagnostics work, right? So when you make an antibody, a monoclonal antibody to target something, traditionally, you'd think, well, I got to target a protein. That's the only stuff that I can access, right? If I'm going to make a CAR T cell, it's got to target a protein. That's going to be on the surface of the cell. So one of the things that we're very excited to consider, right, this is the proposition that we're bringing to the field, is maybe there is RNA out there. Maybe they are accessible to therapeutic targeting, to diagnostic detection. And what would that mean? Why are cells doing it? And while we're figuring out why, we're also thinking a lot about, well, we know it's there. So what if we start targeting it, right? Is there a way that we can actually build new drugs that target this biology that might be better anti-cancer agents, or that might explain why people develop autoimmune diseases, these kinds of things? So there's a lot of very deep biological questions that we've been asking, which will take many, many years to figure out. But there's also a lot of empirical work that we've been doing that's trying to bridge it to the clinic faster than I would have expected, but is actually quite exciting.
That's really cool. Is this all cells or just specific types of cells that have the RNA branches?
Yeah. So the cool thing is that every cell does this, but in a little bit of a different way. And just as an example, the paper will actually be out in a couple of days, I think. But we found that there are key proteins, very specific proteins that bind RNA. Their name is an RNA binding protein, sort of a very simple name because that's what they do. Those proteins shouldn't be outside of the cell. But we found that cells actually do also put those on the surface in addition to some RNA. And we looked across a bunch of cells, and cells are always doing this. There's a little bit of RNA, a little bit of RNA binding proteins, they're near each other. But the crazy thing was that which RBP, which RNA binding protein, matters for your cell state. So if you look at a healthy cell, you might have the RBP1 and 2. But if you look at a cancer cell, RBP6 is really, really high.
Mm-hmm.
That's a fantastic observation for cancer, because what that means is that's a marker, very specific. So what we were finding is that all cells are doing this biology. They're bringing RNA to the surface. They're They're using it for some role, but which ones and how much, very, very cell type specific, very, very cell state specific. And so our lab has a lot of expertise in the cancer biology field, and so we've been thinking a lot about cancer, probably true in other diseases, but our first sort of like therapeutic foray is into cancer therapeutics, where we've been thinking about, like you were saying, which ones are different on different cell types even, and then can it be disease specific? So we found basically one of these that's super disease specific, and it's really specifically expressed on cancer cells, which is quite nice.
That's really cool. You should talk to Alex Murphy. Yeah, I don't know if
Please.
you. Yeah, but
The name sounds really familiar, but I can't remember.
Yeah, I'll intro you after the show. He's at Novartis and literally in Cambridge, right? So
Oh, nice.
at the end of it's like next to the MIT. You know where it is.
Probably near NIBR, yeah, or he's probably part of NIBR, the research institute, or somewhere around there. Yeah, they have a huge set of facilities there. Yeah.
Yeah, and so he's the director of strategy for their neurodegenerative diseases. So like
very cool.
Alzheimer's, dementia. Anyways, but he was on the show talking about it.
Yeah.
Yeah, I'm sure he would be fascinated to learn this, and then also I'm sure that maybe they can kick you some money for your lab.
Yeah, well, the neat thing is that we actually have a lot of interest. I mean, just in this very specific example, but we have a lot of interest in neurodegeneration because a lot of the proteins that we've been studying are in some cases actually causal, right? Mutations in them, changes in them cause LS, they cause Alzheimer's, these kinds of things. But the imagination for what they do has always been that they're inside the cell. And so, unfortunately, my lab is not yet 100 people, so we can't do everything. But we've been thinking a lot about what are the key cool examples to go after. And neurodegeneration actually is quite interesting because perturbations, changes in these are already known to be causal. And so if we have different places to study them and different possible activities for them, it might exist. We don't understand everything about Alzheimer's. It's a very complicated disease. So if we could add a little piece to that puzzle, that might be very cool. Yeah, it's one of the things we've been thinking about.
You just spoke it into the universe. It's not 100 yet. It will be shortly. I actually have a few folks I'm going to introduce you to. But that's incredible work, and it's very exciting.
Well, I'm biased, but of course I think it's quite exciting. Yeah.
Alex or Ryan, sorry, I would love to ask you. Of course, moving a little bit back into life and just a little more generic stuff, like. What have what are some of the big things that you've learned over your career that have led you to this moment or, these big discoveries that you're having?
Yeah, interesting question. So I think I've been very fortunate to be around and be trained by very both smart and kind people. And actually, I think I really enjoyed my time at MIT as a preface to this. It's something that I didn't exactly distill maybe until after I had left. But I think my experience was that like in high school, I thought I'm pretty good at a lot of things. And then I got to MIT and I was like, wow.
I'm not good at anything.
Wrong, right? I thought I knew how to do math. That's wrong. That's basically completely inaccurate, right? But I actually think it's a very important lesson that I think about a lot, right? Like, you can be good at one thing and basically terrible at everything else. And often it's almost certain that like someone else is like way better at you than anyone else in the world, like in all the domains, right? I had a very specific memory of like taking a whole week to do an 1801A problem set and like asked someone and they were like, I had finished it all. And I was like, I haven't even gotten past the first one problem. And I was just like, I just don't know how to fix it. But that was actually very important for me because I think it made me realize and sort of carry with me the fact that working with people is really important in my undergrad time, also in research, but especially as a graduate student and as I come now, the collaborative effort, right? Bringing in people who are very smart, very skilled, have some different expertise. This has really accelerated all the stuff that I've done. I kind of, in graduate school, learned how to be a really deep expert in one aspect of RNA biology. And then my strategy was, I will go talk to as many people as I can, get excited about a bunch of things, and try to help and learn their problems, right? Learn what they want to do, give them a piece of information, a little, like, do my assay, right? And then learn about that, be part of that. And that was actually really fun. I learned a lot more faster because, when you're helping someone, they want to tell you more. If they can get something out of you, it's a little bit more beneficial. And so I think a couple of those things that brought together is like, it's okay not to be the smartest person, but if you have a thing you really care about doing, like focusing on that, and also working with people, right? I think like I've seen that sometimes it feels like that, you're only rewarded if you somehow did all of the things. And then you have to be the deep, full expert on the entire thing. And it's not really that way in reality. I think in life, it's much better, and actually things move much faster when you work together. And so that was true when I was training. And now, with my lab and people in my lab, I try to find ways where, again, people train, they become the expert in something, but we're working together. We collaborate with labs, we collaborate within the lab, we collaborate with labs here in Boston. And then, of course, actually throughout the world. I think we have kind of like active collaborations now with a group in China, a group in Sweden, 4 groups in Japan, or 4 groups in Germany, 1 group in Japan, and others. But like, this is just, and I guess my lab is not even that old yet, but we go wherever, right? And it's very satisfying to find the person who cares deeply about the thing that you care about and work together quickly to find the answer. So I think those things have helped me, move the questions that I have, move the ideas that I have faster to maybe get here and think about some of the stuff.
It's such a key point, right? And when I work with folks, I always get them either through feedback or through their own understanding of themselves. But to get to that, what is the special things that you do that other folks don't? Because the more time you spend in that, the more you're in your zone of genius and the more like great things that come out of it. And often, like, I'm working with a lot of like startup founders or executives. It's like, if you don't have an EA or you don't have a system where someone helps you with booking and scheduling, or your like email, or like, those things are such basic things that, like, for not that much money, you can take care of it and it frees up a lot of your time. And then for folks, like, I've worked with folks who are Bell Labs fellows, right? Like,
Yeah.
your level of research. And it's like, I tell them, do not, like, turn off your phone, turn off your Teams, turn everything off, and like spend time like either reading research papers or doing your work or thinking about your problem. Like, get out of this constant stuff, because otherwise you can't even think.
Yeah. Yeah. It's funny. Well, a couple of things are funny there. Like I actually turn most of my notifications off. Um, I don't have like time limits on any of my apps because I just sort of have pretty good self-control and restrictions. But yeah, everything flashing through is like way too distracting, right? But I was just thinking when what you're saying actually is, it's sort of funny because even for my lab, I still do all the organization and all the administration. So I don't have a lab, I don't have an admin. I actually do all the lab ordering and organization and stuff like this. Probably at some point I'll offload that.
Ryan.
It'll be nicer. But I have a very like detail-oriented sort of view on things, so I end up just kind of doing all those things quickly, but then I can put them out of my mind and think about stuff.
Yeah, I would totally recommend you get somebody to help you with that. And you can also, like, you can find somebody who's very detail oriented too, right?
Yeah.
So I'm not saying just like give it to anybody, but you can recruit and find somebody who is, a perfectionist, a very tight person, and then you train them in the way you like it, and then they go and execute, and then you can spend time leading your group.
That is on the near term horizon. But yeah, it's been interesting because actually I actually only ever trained in very well established labs, like extremely, extremely well established labs where the way I describe it, I think other people describe it as like there's sort of like a machine. It has inertia. It's got a process. It exists. It's fully built. But when you're starting a lab, that doesn't exist. There are no graduate students that have already been trained and graduated. There's no ordering system. There's no strategy for training people on which instruments to use and how to design experiments and sort of things. So it's been very interesting to do that de novo, right? You get advice from people, but kind of the cool thing about academia is you get to do whatever you want, right? But that also means it's completely unique in a variety of ways. And usually you'd only do it once, right? So you don't get to know that you did it the most optimal way, or you've tried it multiple tries and like actually the third version was like perfect. But yeah, it's sort of I think it's smoothing out now, and people in the lab are sort of becoming more independent and training themselves in this sort of thing and evolving in I think a really nice way. So I think it's been fun to watch that over the first about basically 4 years now.
Yeah, and those things, like you mentioned, you only have to do it once, right? But anytime I get folks to write processes or procedures or these things, I always think of it as basically an experiment, right? It's like you're going to try something, and the person who goes through it, they're going to notice things that they think would be better. And so you give them the opportunity to put that feedback back in the system and then improve it over time. And it's no different. Like it's literally the scientific method, right? Is we're continually improving in the same way.
Yeah, a nice iterative effort there.
So I guess, Ryan, how have you been navigating putting all of that together? Like, how do you describe yourself, your leadership style, and bringing everyone together?
Yeah, it's a good question. I get asked that actually by some graduate students, and I always wonder like, what does that mean? What is the answer that they want to hear?
So I don't
Well,
want you
because
to
I
give
think
me
it,
the answer that you think that I want to hear. I want to hear what the real answer is.
well, it's not that I have multiple answers, but I think that, you know, sometimes language is not precise. And so when someone's asking, what is your leadership style? What is your mentorship style? They're asking like a different question, but those are the words that they're using. But I think that actually the way that I think is productive, the way that I have approached setting up my lab, right? Like it's basically like what I always joke is like a poorly funded startup company, um, is this sort of blend of having a vision, right? Having a set of things that we want to accomplish or that I want to accomplish, right? That's the goal of the lab, right? What are the, because it's a scientific lab, what are the scientific questions we want to address? What are the systems that we study? What are the ways that we ask questions? And I tell people that some of it people can read in papers, and then we talk about that stuff, whatever. And then usually it's important that I actually then ask the person, the postdoc that's coming in, the graduate student that's coming in, whatever, what do they want to get out of it, right? I think that my view has always been that, it's sort of like the vectors need to align. You won't be overlapping because you're not the same person. And like I distinctly have different goals than any other person that would come to the lab. But we need to sort of like align and know that the directional effect will be the same. And so there's a little bit of like, I want to go this way. And like you also have some interest and want to go this way, but like let's talk about it and get it so this way, so that at the end, you'll feel productive, you'll feel like you learned something, have accomplished something related to your trajectory, right? And so I always imagine it like aligning vectors is sort of like, and could I describe what a vector is mathematically? Probably not. But in my head, this is a great framework to describe what that is. And I think it happens over the course of a couple of discussions, and I write lists out, and we go back and forth on different projects and ideas, and sort of test things out. In biology and many things, we're not always right, so we might come up with a cool idea, we try it, doesn't work, or something like that. So there's a little bit of processing there. But I like this idea of what do we know, where are we starting, where do we want to go, and then very specifically with students or trainees at the various levels, like what do they want to do, what is their vision, and the vision can change, right? But what do you think today? Do you want to run a lab? Do you want to start a company? Do you want to work at a company? Do you want to be a consultant? Like whatever it is, right? It's very helpful to know, right? How long is that journey going to be? What are the steps along that will generate value for you and your training process? So I think that this is like how I describe it and how I try to communicate to people that come to the lab. And then, every person's also different. Everyone hears that differently. Everyone imagines that a little differently. So it evolves in sort of a unique way, I think, with each person. But that's sort of the way that I like to think about it, because it's not really, I don't think it's useful to have a rigid framework, because like every person is different. And I think the utility of having a system like academia is that it really is customizable. You can do whatever you want, and you can try to get out of it whatever you want. So having an arbitrarily rigid structure is like not efficient, I think. And so, yeah, that's sort of the way I think about it.
It's a great way to think about it. And I tell everybody to do this, academia or not, right? Because ultimately, the hardest part is some folks won't tell you. If, like, let's say you're their boss, right? And you're saying, hey, where do you want to be in 5 years? And they're like, well, I want to start my own company, right? Like, they're not, most of the time, they're not going to tell you, at least up front at the beginning, right? But it's aligning, where are you going? Where do you want in your career? What are your values? What excites you? What interests you? What are you good at? And then if those match, then you're right. The vectors align and bang. I mean, of course, they'll be slightly, but you can adjust, right? But then bang, you have a partnership. If you don't, like if the person's like, well, I just, I want to do, I don't know, some drug development for, some company. It's like, well, okay, then that's not here. And that's totally cool,
That's a
right?
great idea, but like not helpful.
Yeah. And it just doesn't work out. I mean, it's not a bad thing. It just is. And you're right. People, they change over time as they learn and as they grow and as they develop. And that's normal. And so it's never, and this is something that I think some folks get wrong, is they think it's like, oh, we're in it forever. And it's like, for the most part, no. Like, a couple of years, maybe 5 years. Sometimes it will be 10, 20 years, right? But you can't, like, when you're walking in and you're starting to work with somebody, it's like, don't have that expectation it's going to be forever, because that's going to hurt you.
Yeah, and I actually, um, I don't know how much it gets through, but sometimes in the class I teach an undergraduate class at Harvard. In the beginning of the class, I have like discussions about the class and like the goals of the class or whatever, and then um, I think I forget exactly what I write, but basically like one of the last bullet points is like um, spend time. Like I'm going to ask you at the end of this class, what do you want to be doing in 5 years? Like, what is your vision for the next 5 years, right? And it's like undergrads, like they're like sophomore, junior, seniors. But I think it's actually perhaps helpful or interesting because I think a lot of times even academic training doesn't really reward long-term thinking. There are a lot of tasks to do right now. There are a lot of requirements to get done. There are a lot of like short-term things that you have to do that are feeling productive or are productive, but are irrelevant to the actual trajectory that you're going to go. And there is no requirement. There is no checkbox that says I have formulated a really careful vision for what I want to do with the training that I'm getting here and how that will help me to do the next thing. There is no place for that, right? It's just like maybe you did that on your own time and that's great. But I actually think this is very important because I feel also like, and I describe this to people in my lab, but like usually irritation, disgruntlement, I think happens because like incentives are misaligned or the agreement is somehow broken down and then people don't think things are happening correctly. And if you don't know why you're doing something, very easy to have that misalignment, right? And so if you don't know why you wanted to come to grad school, if you don't know why you wanted to do a postdoc, if you don't know why you got this job, whatever it is, but you did it because that was the opportunity, you did it because that was just the next step, then you can get there. If it becomes hard or something goes wrong or whatever, then you're like, oh, like why is this so annoying, whatever. And so it is hard to build that in, and each person has a different interest and capacity to do that. But I think it's also very important to consider, right? Like, why are you doing something? Not even in a negative way, but like it's an opportunity to kind of think positively about it and be like, I have these goals. I will accomplish these things, and then when I'm done, I can leave. Right? And it actually helps you. Maybe you thought it was going to take 5 years. It turns out you accomplish those goals in 3 years. Boom, you get to go. Go ahead, go do your thing. So there's another thing that I think about a lot with respect to people in the lab.
It's brilliant, right? It's, and it's, a lot of folks don't have that 5 year. And okay, if you're listening to this, right, your 5 year vision's not going to be right. Let's just be honest here.
Yeah,
5 years ago, I was a depressed engineer living in Edmonton, Canada, and I never would have guessed I would have became a coach. I never would have guessed that I would have done a TED Talk, written a book, moved to Costa Rica, got married. None of that I would have ever thought of, right?
yeah. A lot of
So,
changes in 5 years. Yeah,
yeah. But as you start to learn who you are, that's the closer you can get to that 5 year vision. And then it can be these deliberate steps. And so along that journey of, starting to do the work, I started to get more clear on things. I wanted to do speaking. So then when it came up, I hired a speaking coach to help me get TED. Then I did that, right? And then it was like, I should write a book. And then you start getting help writing a book, right? And like, then you can start choosing. And I think what you're so smart about, Ryan, is like in undergrad, if you would have said that to me, I would have been like, I guess I want to get a job, right? And I want to make some money or something. But I think the more clear you are on your goals, the more obvious decisions are. And then you're not just like left up to life going, oh, well, this person offered me a job, so I'll just take it because it's the best thing I have right now.
Yeah. Yeah. And like you said, like, I don't expect anyone in the class to like have thought about that, right? But it's sort of just like throwing it out there and being like, oh, I've never heard that question before. That's an annoying question. Like, what would I even say to that, right? And like you kind of like try to think about it a little bit, and then maybe they think about it later.
What would you say? Where do you want to be in 5 years?
Hopefully here, physically, in Boston. Yeah, I mean, I think that for me, I think like, yeah, I guess there are different ways to look at that. I think that at the level of the lab, I would like that machine to exist, right? I would like there to be a system where people can come and train and learn the biology that we're doing and pick a new piece of it and figure something out for themselves and then go on and do their own thing. And whether that own thing is their own lab or a company or something else. I think right now we're building it, right? It's coming together. But I think people in the lab are still like really actively contributing to figuring out how that works. And it's a lot of effort. So hopefully within 5 years, let's say that's like a more systematized activity. It'd be really cool, I think, if some of the work that we're doing is translated in some way, better disease connection, commercialization of some of the discoveries that we've been making, try to better understand, what is the disease relevance of the basic science that we've been doing. I think that would be really exciting and really rewarding. Yeah, I think those are like the things I've spent most of my time on, right? Like, how do we learn more faster about the questions we have? And then what are their relevance? Right? I could study this forever internally. I think it's just cool. I just think the answers to the questions to me are very interesting, right? So I don't care. I almost don't care. But on a practical level, it's actually very important and very interesting to know. Will they matter, right? Will it be interesting? Optimistically, we can find some of those things out, but obviously the purpose and the sort of utility of the way academia works, the way science works, is like we'll share these things, we'll be publishing these papers, and then someone else will see it, and they'll be like, actually, I know exactly how to test that, right? I know the precise thing to do in this other disease model. So the 5-year horizon is actually quite exciting because it is enough to turn that crank a couple of times where some of the first work that we've done will be out there, people will read some of it, they'll be like, oh, actually, I've heard of that gene before, we have a way to modulate that or change it, or I've seen a disease where that's different, I'll go test it, and then they can do their work, and then they can get their stuff published, and then we can see, right? So I think that'll be actually a really exciting kind of turn of the crank, right? The conceptual crank there. And so hopefully we'll see some of the initial ideas that we have, translated more broadly. We'll see.
Amazing. I love that. And of course, so how can the rest of the MIT 2010 support you in achieving that mission?
I haven't thought about that. Support science in general, I think, we rely, right, on like a very diverse set of societal features, right? We need people to come and train. We need money, whether that's federal, state, philanthropy, industry, right? I think one thing that I've been thinking a lot about lately is how do we better explain what we're doing? We're sitting here finding data, finding results or whatever, and then like, does anyone know about it? Does anyone care? Like, I think about this more generally, given stuff that's going on, I think, in the world, but the dollars for biomedical research, the dollars generally for any type of R&D are sort of like comically small with respect to general spending, but they underlie basically everything that we do. How we're talking now, backing up in the cloud, all of our phones, the food that we eat, how it's manufactured, how it's moved around, all of that is supported, right, by science. I think the cool thing about science is that it's like immediately and generally true for everyone. My RNA works the same as your RNA. That's very cool, right? But like, because it's so fundamental, because it's so boring in that way, you don't have to think about it, which is great. You don't need to remember every time you're making a new protein and that protein is doing the thing, right? But what that means is people, I think, forget about science and what it's doing and these sorts of things. And so I do wonder, like, how can we better communicate that stuff to everyone that's using, right? That's the beneficiary of those things. I don't know the answer. I don't know the answer to that. But I think that's something that I've been thinking about is for people in different domains, right? For people in finance, for people in consulting, for people in science or government or policy, like, I'm out here just trying to ask questions about RNA, right? I think it's very cool, and we think that it probably will be perhaps interesting and important. But then I'll write my paper, I'll go on with my life. Maybe I'll have I'll be on a podcast. I'll talk about it a little bit. There might be a news article about it. 10 people will read it or something like that. Um, yeah, I wonder how we can better promote that stuff, right? And excite people about that. Because broadly speaking, the MIT community especially, right, does incredible work, and kind of it's very exciting internally. And then I think no one, round down level, but like at an absolute level, very few people think about that, which I think is technically good. You don't want to have to think about all those like underlying functions, all those underlying mechanisms and processes, but it means that when you forget about them, you don't remember that they're important, and you don't remember that they could help you, or that they are helping you, right? So, yeah, I don't know. That's something I've been thinking about, but it's just like sort of a musings. I don't have a good, I don't have a granular need, a granular ask.
You're coming into my world. I can help you with that. Let's not talk about that, folks. So, of course, obviously, if you're interested in the research, if you're interested in, Ryan's going to be at the reunion, I'm assuming, just because you're in Boston. So it's not
yeah, I'll be around. Yeah.
But yeah, you can definitely hit up Ryan. I put his LinkedIn in the podcast notes as well. And, reach out, chat with him. There's obviously incredible science here. I'm going to make some connection to fellow 2010s probably this weekend. Ryan, I always ask this to everybody that I meet. Is there any person or type of person that would help you to meet them?
Say it once more time. I just broke up for 1 second. Is there any
type of person, or like you could say like somebody who works in pharmaceuticals, you could say like an investor in health, or you could say, I don't know, like, someone who runs a lab in whatever, but a type of person that would help you accomplish your goals?
Good question. Yeah, I think that thinking very specifically about some of the stuff that we're doing, I think people that commercialize drugs, spin technologies out from an early stage, thinking about the transition out of academia of ideas, whether that's straight into pharmaceutical, level companies, whether it's independent startups, this sort of thing. I think that's a very interesting thing for me to learn more about and hear about, because everyone has different interests, risk profiles, intersections of those things. Understanding what is this, what is the state, what is the scope of things that people are interested in doing now, what things are moving forward and what are not, would be very helpful and could help steer some of the ideas that we're working on, may give me new ideas, this sort of thing. But probably that would probably be exciting. Yeah.
Amazing. Folks out there, if that sounds like you, or somebody who does that, definitely reach out to Ryan, make a connection. Ryan, is there anything else you'd like to say to the 2010s out there that you haven't said already?
I'll just reiterate that it was great to learn that I was not the smartest person. And I really do think that is a very valuable lesson. And I actually wish that, so I got that being a 10. I do genuinely wish, though, that more people in the world could have that experience. It's not a bad experience. It's actually very important, I think, for people to understand that they don't have to have every right answer. They don't have to be the smartest person. It doesn't make you dumb. It doesn't make the things you want to do worse. It can really help you think more clearly and get help and and build up a team, right, that is actually very productive. And so I thank MIT and the class of the 2010s to help me learn that very quickly, but also over the 4 years. Yeah.
I totally resonate with this, right? So I remember being in 801, the first class of 801, and the professor puts up the outline, and momentum was week 9, and I'm sitting there going, great, I'm good till week 9. And then they pull out like a double integral like 5 minutes later, and I was like, oh, I'm not good at all.
Yeah, exactly.
Yeah. So anyway, I'm sure folks are laughing and they're listening right now. But yeah, it's incredible. Ryan, such a pleasure having you on the show. I mean, I'm really interested on how your work and where it's going to go and all the things it's going to lead to. I'm definitely not in the biology space, but you've piqued my interest, and it's incredible to see someone making this kind of impact, because it's awesome. So thanks for joining us. Thanks for sharing us. And I'm looking forward to meeting you in person and seeing you soon in May.
Yeah, it's been great talking, and I'll see you basically in a month.
Awesome. Thank you so much. Everybody who's listening, thank you so much for listening. Of course, if you're looking for executive coaching, or if you book speakers or know somebody who books speakers, definitely robkalvarowski.com. You can reach out to me or rob@robkalvarowski.com. And then, of course, if you like this episode, share it with the folks around you. And if you're watching video, of course, that's Mowgli in the back, but she's come to say hello. So everybody listening, thank you so much, and we'll see you next week.